It's my great pleasure now to introduce Larissa Arning from the Ruhr University of Bochum, beautiful university. She will talk about non-pathogenic CAG tract and gray matter microstructure in the human brain.
Yeah, thank you very much for inviting me to share our results. I begin with a neurodevelopmental hypothesis of Huntington's disease, which suggests that the CAG repeat expansion is only the pathological extreme of a more general mutational process that also contributes to normal brain function and development. And as we already heard in the nice talk this morning from Elena Cataneo, the CAG repeat in the Huntington gene may have been selected throughout evolution, and small increases in its physiological length influence neural functions of Huntington. In the figure underneath, adapted from Van der Plas et al., this figure very nicely represents the hypothesis of the effect of the entire CAG spectrum from low to high, with repeats below the disease threshold. It's suggested that increasing repeats being advantage, for example, resulting in better cognitive performance. In contrast, repeats above the disease threshold are disadvantaged. So far, not many studies have analyzed the effect of the physiological repeat length in the Huntington gene. Different studies have found an effect on brain structure or better cognitive performance with increasing repeats. But on the other hand, also a CAG repeat length-dependent risk of lifetime depression has been described. The aim of our study was to expand on these studies, and we took a comprehensive analysis in over 500 healthy adults. We assessed the broad cognitive battery, examined the macro and microstructure of several MRI measures, and controlled all these measures for an effect of the CAG repeat length. I'll give you a brief overview over our methods. This sample cohort has originally been selected by the Department of Biopsychology in Bochum with the intention to find neural correlates for intelligence and general knowledge derived by imaging data. The cohort comprises 533 mentally and neurologically healthy participants. With a mean age of 27, the cohort is quite young. That's because most of the participants were university students. Analyzing the cohort was a CAG repeat length revealed for the long CAG repeat a mean length of 20. The comprehensive cognitive testing was based on different tests. First, the Intelligenzstruktur test. This comprises various types of mental test items to measure multiple facets of general intelligence. It's comparable to the Wechsler Adult Intelligence Scale. Then the Bochumer Matrizen test. This is a non-verbal test of inductive and deductive reasoning. This is comparable to the Raven's Advanced Progressive Matrices. Then the Bochumer Wissenstest. This is an assessment of general knowledge, comprising the facets of humanities and science, and at least the Zahlenverbindungstest, which measures general intelligence by the assessment of information processing speed. All imaging data have been acquired at one study site in Bochum. T1-weighted images were required, and for the purpose of analyzing NODI matrices, a diffusion-weighted scan with multiple shells has been performed. And for the purpose of brain segmentation, We used the Human Connectome Project's modular parcellation, which results in 180 cortical areas per hemisphere, as well as 8 subcortical areas. In the end, a region-specific multimediator analysis was performed via elastic net regression for each combination of brain metric and CAG length. So I come to the results. The results for the comprehensive cognitive testing, and to make it brief, there was no effect of the number of CAG repeats in Huntington below the disease threshold on any test outcome. And now there's the results for the comprehensive imaging testing. There was no effect on surface area, cortical thickness, volume of the cortex, volume of subcortical regions, volume of white matter, no effect on white matter microstructure, and I didn't mention it before, graph analyzers have also been done. There was no effect on structural brain network efficiency, no effect on functional network efficiency. But what we found was an association with NODI metrics associated with gray matter microstructure. So NODI, what does it mean? This is a neurite orientation, dispersion, and density imaging. It's a diffusion-based MRI technique that allows for the in vivo assessment of brain microstructure at the level of neurite morphology in gray and white matter. But when we look in the literature, most studies who performed NODI analyzed it for the white matter. NODI derives microstructural parameters indirectly based on the diffusion patterns of the examined tissue and can therefore distinguish 3 types of microstructural environments in the brain. Thus are the neurite density index, the orientation dispersion index, and the isotropic volume fraction. And in our results, the neurite density index showed a relationship with the physiological repeat length in the Huntington gene. I give you a brief overview over the areas associated with increased neurite density, and they appear to be distributed all over the cortex. And to give you a very rough functional orientation, one can say that part of the areas with an increased neurite density are associated with visual areas, but they are also localized in dorsolateral prefrontal cortex, associated with memory and cognition, and the orbitofrontal cortex associated with emotion. These are the areas associated with decreased neurite density. Here, the strongest effect is seen in the primary motor, somatosensory cortex, but we see also associations again with visual areas and emotion-related areas. Looking at the subcortical areas, there we see decreased neurite density in the globus pallidus. So what could be the possible interpretation? Gray-Medi neurite density is a robust indicator of cortical myelination. Therefore, the strongest contributor to variation of neurite density across the cortex is likely the density of myelinated axons. Since genetic factors account for approximately 50% of the observed individual differences in mean cortical myelin, the physiological CAG length in Huntington may therefore play a role in the genetics of cortical myelination and thus explain individual differences in intercortical myelin pattern. Since the cortex has undergone significant evolutionary change across species with an expansion of the lightly myelinated regions in humans, decreased neurite density might enable greater plasticity. In contrast, decreased neurite density may also represent underlying myelination abnormalities in these regions that lead to cortical pathogenesis once the repeats expand behind a certain threshold. So to have a brief look at what is known about the cortical pathogenesis, we see in this heat map the somatic expansion of different brain areas. Most of these areas have already been part of the publication from Pinto et al., but we extended these analyses to further areas, all marked with an asterisk. And what we have learned is that the somatic expansion is highest in the cortex, then followed by the striatum. And what is new is that a region, a high phasor region like the corpus callosum, genu, and splenium also shows a high rate of somatic expansion. Other areas, or other phasor areas like the optic nerve and optic tract, they only show a low somatic expansion, but they belong to the diencephalon and not the cortex. Furthermore, we wanted to compare these regions with aggregates. We looked at the corresponding areas and the aggregate density. And again, here we see the aggregates are high in the cortex, but directly comparing the areas, you can see that it doesn't really or isn't the same pattern, especially when you look at the visual cortex, for example. And the same is with the neurite density in healthies. The areas associated with increased and decreased neurite density also do not really match to this pattern. So in conclusion, in the context of rapidly developing Huntington lowering therapeutic strategies, basic research on the function of wild-type Huntington needs to be continued to optimize the efficiency and safety of future therapies for Huntington's disease. To fully understand how mutant Huntington might affect brain development, it is important to consider the exact function of the normal gene. We showed here that an increasing number of Huntington CAG repeats below the disease threshold was not associated with improvements in cognitive performance, but we have to say that this was a very homogeneous cohort, young and all university students. So it would be interesting to look in bigger cohort, a wider cohort. But what we showed is an increase in its physiological length might influence the density of cortical myelinated axons. However, it is unclear if this is an advantage change in the function of the developing brain. Therefore, our results are merely correlative and therefore need to be validated. I want to thank everyone who contributed to this work, especially Elizabeth Petras-Pavish, who was a main contributor of most of this work here. Thank you.
All right, thank you very much, Larissa. We have time for a couple of questions. We have a question down here. Yep, yes, I think we'll need to wait for the mic.
It was, they were checked or asked for any mentally or neurological disease. And of course, you don't know it exactly, but it was, as far as it's possible, it was checked that they are healthy. And the first, it was a work I mentioned that found a nonlinear association with CAG repeat lengths and earlier depression in healthy. This was a work from literature.
Great. I think we maybe have another question in the back here.
So, question regarding this nonlinear effect that you mentioned. So according to the neurodevelopmental hypothesis, there should be an optimal CAG repeat size after which increasing CAG repeat size is considered to be detrimental. So I wondered whether you also specifically assessed any nonlinear effects within this normal range of the Huntington CAG repeat size.
No, not yet, but that's, that's an idea because of your paper with depression.
Yeah, okay, thanks.
Quick follow-up to that same question. So as repeat expansion numbers trend towards 27, maybe 36, do you begin to see higher and higher NODI associations?
You mean patients in the higher end of the physiological length, or?
Yeah, still physiological expansions under the disease threshold, but what happens to your brain imaging data when you analyze the NODI?
I didn't get it really.
Okay, I think in the interest of time, we'll have to move along nonetheless. But thanks again,
Okay,
Larissa.
sorry,
You can always grab the speakers in the sessions in the breaks.